look at here is the pour plate technique, and this technique kind of sits in between the aforementioned two techniques of the streak plate and the spread plate technique. And this is used for quantitative and both qualitative purposes for both enumeration and isolation, and this is because the colonies kind of form on the surface and both within the agar. The principle between the pour plate technique is that microorganisms are mixed into the molten agar before it solidifies, and this creates a 3D growth environment. It naturally forms oxygen gradients, so organisms that can tolerate lower oxygen can still grow deeper in the agar. Procedure-wise, you add a measured volume of the diluted sample onto the sterile petri dish, and then you pour molten agar that's cooled to around 45 degrees Celsius, and you gently swirl the plate in order to distribute the cells evenly and let the agar solidify before the process of incubation. This method is best used for low microbial load samples, and it's especially useful for microaerophilic and facultative anaerobic organisms because not all colonies are forced to grow at fully oxygenated states on the surface. In terms of the limitations, first limitation is that the heat from the molten agar can reduce the viability for heat-sensitive cells, and subsurface colonies are harder to observe and count, and the colony morphology can look a little bit different because the growth conditions inside the agar aren't necessarily the same as on the surface.